just want to say thank you for inviting me to speak and present the results from the evaluation that was done with the DevTox Quick Predict assay. I'm just going to go over a little bit of background on the assay so you can kind of understand what the data is that we're going to look at, and then an overview of the results that we received. And so Stemina's assays are built around using human pluripotent stem cells, or cells that are derived from them. And the idea is that we want to use a human cell line to try and minimize or eliminate differences that are due to species, differences in species for toxic response. So the DevTox assay that we use and that I'm going to talk about uses undifferentiated pluripotent stem cells. And we do all of our treatments keeping them in that undifferentiated state. And the reason that we do this, and we chose to do this, was because we didn't want to go down any specific lineage, which could then limit our specificity for our sensitivity to that specific lineage for an effect. The assay does work with both embryonic and induced pluripotent stem cells, but everything that we really do now is with the induced pluripotent stem cells, and that's what we're taking forward for qualification with the FDA. And then we also have a cardio assay that looks at using human iPS-derived cardiomyocytes. And keep in mind, as I'm talking, that the idea that we had when we developed our quick predict assays is that we wanted an exposure-based prediction of toxicity, and we use a targeted metabolomics platform. So everything, as we were designing it, everything was based around the fact that some toxicity is a function of exposure. So the assay is optimized for use with human embryonic and induced pluripotent stem cells. And to identify our prediction model, we started with a training set of 23 well-characterized compounds that had known human developmental toxicity outcome. And we exposed each of these cell types to these 23 compounds multiple times. And we were looking at the secretome. So we wanted to see what is changing and how these cells interact with the media. We're collecting the spent media and looking at what are the cells taking up, what are they secreting into the media, and how does that change when you treat them with one of the developmental toxicants versus treating them with a non-toxic type. And through doing this over multiple replications and many years, we built a number of very complex models that had good predictivity, but they also contained many things that were unknown and couldn't be confirmed as a specific metabolite. And so we wanted to really narrow that down to things that are known and, try and simplify the assay as much as possible so we can give a quick response to our clients as to whether or not a compound is going to be developmentally toxic or not. And so through doing this in multiple iterations and looking at pairs of metabolites and what was most complementary and predictive, we found that if we used a ratio of ornithine and cysteine, so if we looked at the effects of these two metabolites, these two amino acids, we were just as predictive as those much more complex models with unknown features. So why are these two important? Ornithine is secreted by the cells into the media. It most often will decrease with toxicity, but there are certain cases where it can increase. And that is large. Basically, we've only seen that with things that are in the retinoid family, because they do have a very specific mechanism that's been shown in multiple studies where they can inhibit the enzyme that breaks ornithine down. And biochemically, ornithine is critical for the urea cycle, polyamine synthesis, and pyrimidine synthesis. Cysteine, on the other hand, is a media component. And when the cells are happy and healthy, they're consuming the majority of the cysteine that's in the media. But when you treat them with something that is toxic, you're going to see an increase in the cystine. So what that really means is that the cells go from using all that cystine from the media to not using it. And its main function is in the cystine-cysteine redox cycle, which is an indicator of oxidative stress. But there are a number of other pathways can ultimately impact cystine uptake in the cells, leading to a positive effect. And both of these are key amino acids that are required for normal proliferation and differentiation. So you can imagine if you're thinking in vivo or down the line, if you disrupt these processes by disrupting the metabolism of these metabolites and the production of these metabolites, that's going to disrupt normal embryonic development. The process that we use is we'll take the cells in their undifferentiated state and plate them into a 96-well plate. And we'll look at 8 concentrations of each chemical or each sample. And that concentration range is going to be based on, discussion, the chemical type, discussions with the sponsor, the solubility, really all the, we'll take into account all the data that's available to us. And then every plate's going to include positive and negative controls to make sure that everything in the assay is performing as it should, that the cells are metabolizing in a manner that's consistent with the predictive model. And we'll expose the cells for 48 hours with daily media changes. So we'll add the treatment, and then 24 hours we'll replace that media with new treatment, and then collect it. And then we'll take that spent media and we will remove all of the proteins and do a targeted LC-MS analysis for ornithine and cystine. And this analysis also includes internal standards to remove instrument variability and et cetera. And because we're looking at that spent media, it allows us to do downstream analyses on the cells. So we always do a cell viability assay to look at, so we're able to compare the results of the metabolism to the cell viability. And it also leaves these cells open for other downstream analyses if there's a need to do gene expression studies, et cetera. And so then we'll take these 2 data endpoints and look at the dose response curves to predict where is a compound going to be developmentally toxic, or does it show an effect. So if we blow that up a little bit, that graph up, and look at it a little closer, like I said, the idea is that toxicity is a function of exposure. And so when we were developing the assay and we were using that training set of 23 compounds, we identified what is the critical threshold or the amount of change required for a compound to be developmentally toxic. And so this threshold is based on comparing the response in the training set to the concentration in humans where developmental toxicity has or has not been observed, in addition to looking at the variability of the data points and making sure that we're not within that noise belt. And so using this threshold, we'll predict from the OC ratio what we call our developmental toxicity potential concentration. So I'll refer to that probably as the DTP and when I'm talking about the results from this study. And this is at what point does the dose response curve for the OC ratio cross the threshold. So it's an interpolated value based on the model that is fit to the data points. And then, like I said, we always look at cell viability in order to compare and understand, are these changes occurring just because of effects on cell viability, which is still important, but it can impact how you interpret the results. And using this DTP, it really kind of gives you your non-toxic and toxic exposure windows. So anything that is lower than the DTP would be a non-developmentally toxic exposure range, whereas anything above the DTP would be your developmentally toxic exposure range. And we've been doing this assay for about a decade now, and we have looked at a large number of chemicals in both cell types. The data here is the accuracy of the assay for pharmaceuticals, agrochemicals, cosmetics, industrial, and environmental chemicals in the IPS cell line. And in the single chemicals, this more simple, obviously, than the botanical extracts that we're looking at, the assay has a pretty consistent accuracy, sensitivity, and specificity of about 86%. So for this study, we looked at the 13 extracts that were provided by the Botanical Safety Consortium, and we tested them using the human IPS cells, so looking at a 48-hour exposure range. And we were limited on our concentration range by the solubility of these extracts in DMSO. So the way that everything is done is we first create a DMSO solution at 1,000 times what our concentration is going to be in the cell culture media, because the cells, we can only use up to a 0.1% DMSO concentration in the media for treatments, because when you go above that, you start seeing effects on cell metabolism just because of the DMSO. So you want to stay away from that. And so there were a couple of extracts that we weren't really able to test at very high concentrations with a maximum of 5 micrograms per mil for the ashwagandha root and 6 micrograms for the kava extract. And so when we looked at the data, we found that one compound didn't impact, and I know that these graphs are really busy and I'm going to simplify that, show more in-depth ones, but what I wanted to do here is kind of show the range of responses that we observed, because there was a lot. One did not impact the OC ratio at all at the concentrations that we tested, and that was the ashwagandha root. And 12 of the 13 did decrease the OC ratio at some point within their dose response curve. But there is a very broad range of effect levels. So we have compounds that were changing it at 0.06 micrograms per mil, whereas something like milk thistle didn't change the ratio, or it didn't cross that, I should say, it didn't cross that threshold. It changed the ratio, but it didn't cross that predictive threshold until you got up to about 89 micrograms per mil. And 10 of these extracts decrease the ratio independent of any change in cell viability. So that means that either there was no change in cell viability across the exposure range, or if there was a change in viability, it occurred at concentrations that were more than 3 times higher than where we saw an effect in the metabolism. And then 2 of them changed the ratio at concentrations where we saw similar to those that we saw an effect on viability. And when you look at the viability, only 4 of them actually were cytotoxic to the human IPS cells within the exposure range that we tested. So I'm not going to spend a lot of time on this table, but what I kind of wanted to point out was when you look through it, we do have that very large range of chemicals or of responses and concentrations where you see an effect. And you can start to kind of see where, if you wanted to do something like a potency evaluation, you can kind of start seeing where things are, certain groups of the extracts were toxic to the cells at similar concentrations, which would have, similar potency in vivo. And so there was really kind of 5 different groups, which stands out a little bit more when you look at them in their figure. Like on the graph, you can kind of see that these compounds the cells are responding to these compounds not only at similar concentrations, but also have similar shaped dose response curves. So the most potent compounds in the assay were golden seal, blue cohosh, comfrey, Usnea lichen, and the Astrolechia Fangfi. Sorry, I haven't had to pronounce very many of these out loud just in my head. Then kava, kratom, ginseng, and ephedra were the next most potent group. Yohimbe was kind of in between group 2 and where the least potent group that caused an effect in the assay, which was green tea and milk thistle. And then the ashwagandha root didn't cause an effect, as I said, but again, we have to keep in mind that top exposure was only 5 micrograms per mil. So as I mentioned that, not only are these compounds grouping together based on their concentration, where they're causing an effect in the assay, but they're also causing different types of effects, which really could indicate different or multiple mechanisms being involved in how the cells are impacted or how the extracts are impacting the cells. So in this category 1, or the most potent group, you can see there's a number of chemical extracts that are causing a biphasic response, where you have these 2 inhibitory phases. Part of that would be caused by an initial response in the cystine, followed by a more striking effect in the cystine when a compound becomes cytotoxic. But as I had mentioned, only 2 of these were actually cytotoxic in the cells. So it could also be that after a certain concentration is reached, there are multiple mechanisms impacting how the cells uptake that cystine from the media. In the second category, and the third category, you see these nice 4-parameter dose response curves that are much more typical of what you observe in our assay, as well as many others. And you can see they're kind of causing effects at very similar concentrations. And then the last 2 groups you can see that are causing an effect in the assay, as well in cell viability, as well as the metabolism. The milk thistle caused this bell-shaped curve where you see an increase in the OC ratio followed by a steep drop-off when the milk thistle becomes cytotoxic to the cells. And this increase that we're observing here is due to an antioxidant effect, where it's actually causing the cells to take up more cystine than the DMSO controls. And then once it kind of reaches that critical level where the cells can no longer respond to the effects and they die, it just drops back down and cystine goes up. So in summary, 12 of the 13 elicited a response that we would consider to be potential for developmental toxicity in vivo, but only at or above that DTP concentration. There was a broad range of potency that we observed in the assay, and you can group those into 5 potency categories. And then, interestingly, there were significant differences where the dose in the dose response curves that indicate that there could be multiple mechanisms or pathways that are being affected either within a botanical extract and between the botanical extracts. So with that, I'd like to thank my team here at Stemina and all of you. And again, thank the Botanical Safety Consortium for inviting me to talk and asking us to test these. I really enjoyed doing this study, and I'll take any questions.
Great, thanks so much, Jessica. So there's a question from Andre in the chat. Do you run qPCRs for trilineage markers to see if the extracts push the cells towards a specific lineage?
We do not as part of our standard assay. Like I said, sometimes we will collect those extracts and do that. But most of the time, the work that we're doing is really, they're not looking for mechanisms. They're just looking for a yes or no. So it's something that we can do, but not something that is standard done.